Are microplastics in the world's oceans being accurately quantified? Hello friends, Jim here. Many microplastics in the world's oceans have likely escaped detection, researchers say. So, this is a lab-generated image on the Raman spectra, shows a microplastic particle within a 100 micrometer area. And for scale, the length of this line segment represents 100 microns, so you can get an idea of the size of it. Okay. Chemicals within the microplastics that are found in our world's oceans and waterways and in drinking water sources is a big concern to scientists and public health officials. A new study that investigated the presence of the smallest particles of microplastics in ocean waters from the Caribbean to the Arctic found that the most abundant and the smallest microplastics in the ocean are not being detected by net tow surveys. Okay, what is meant by a net tow survey? This is basically plankton nets. Now, you can deploy plankton nets by lowering them to a certain depth and then hauling them up to the surface, what we call a vertical tow, and you collect samples that way. You can also tow them horizontally. For example, like a Neuston, which is spelled Neuston, a Neuston tow that is typically used for gathering surface samples. You could also set it so that it sinks to a certain depth and do a horizontal tow in that manner. So you can run plankton tows vertically and or horizontally depending on the desired sampling regime. The research was conducted by scientists in the School of Marine and Atmospheric Sciences, or SOMAS, at Stony Brook University, led by Louis Medina Fowl, and their findings were published in a paper in the May edition of the Marine Pollution Bulletin. The proliferation of microplastics in aquatic systems is dangerous as they can have negative biological effects on living creatures in the sea and on land. Additionally, microplastics can enter the larger food web and make their way up to humans through seafood consumption. Right? Make their way to humans through seafood consumption. Now, for long-time followers, you know I have made many videos on plastics. I've shown videos of larval fish ingesting plastics. I've made videos detailing or explaining how microplastics have been found in human blood. And there's a whole bunch more videos I have made. If you're interested, go to my YouTube channel and click on the video tab, and in the search box type in plastics and it'll all come up if you're interested in seeing any of those videos. The use of Raman microspectroscopy to investigate many samples of sea surface water led to the findings. The laboratory technique is vibrational spectroscopy that combines with a microscope to detect and chemically identify minute particles in environmental samples. The samples are taken from 3 distinct global ocean regions: northeastern coast of Venezuela, Gulf Stream current, which encompasses the Caribbean waters and throughout the Atlantic Ocean, as well as the Pacific Arctic Ocean. That's like Alaska. Standard net tow surveys in oceans detect microplastics as tightly meshed plankton nets sieve out microplastic particles during the tow. When they say tightly meshed, they're talking about openings very, very small. However, viewing ocean water samples under Raman microspectroscopy opens up a new world in defining the smallest of particles. The researchers measured microplastic detection in micrometers. Well, we also have, don't forget, the way microplastics are defined are particles that are like 0.1 micron up to 5 millimeters in length. 5 millimeters, of course, is visible to the naked eye, so it's not necessarily all that micro. Then there's also a category of nanoplastics, which are smaller than 0.1 microns. 1 micron, is 1 millionth of a meter, right? 10 to the minus 6. And that's the size of the common bacterium like E. coli. Plankton nets detect particles mostly from 300 to 500 microns, seldom smaller. Because you have to look at what are you targeting. Are you targeting, for example, zooplankton? Then you're probably going to go for the upper end of this. If you're targeting phytoplankton, you're going to go to the lower end of it and maybe down to, say, 100 microns. But when you start getting into the 1 micron or so range, 1 to 10 micron, then you're looking at what's called nanoplankton. You're looking at the bacteria in the oceans. And admittedly, this is an area of research where there's a lot of missing knowledge. It's just difficult to sample, right? The ocean's a pretty big place. So the size of the plankton net, the mesh size, is determined by what it is you are wishing to sample, what is you wishing to collect and analyze. They'll have limitations. And usually the way these things work is it's kind of funnel shaped for the vertical ones, and it's got these fine mesh, and it directs everything down to basically a collecting object where the net coalesces into, and then you collect that, you pour it off, get your samples, and you treat it, for analysis. Sometimes if you're doing a lot of samples, it's just best to collect it, fix it, and then bring it back to the lab for analysis. Sometimes you have dissecting scopes, microscopes on board ship, and you analyze them in situ. If you got a big enough team, you got one bunch of folks collecting the samples and another bunch of folks analyzing them and quantifying everything they find. And if you're a physical oceanographer, you say, enough of this playing with the bongo nets. We call bongo nets when there's two planktons that are sent side by side down as a pair. They kind of look like bongo nets, oversized bongo nets. And, the biological oceanographers, they got to wrestle those things to get them on board deck, get the samples and this, that, and the other thing, often in, not so great weather, the deck, bouncing and rolling around. Whereas if you're a physical oceanographer, you throw CTD overboard and you sit at your computer and you watch the data downloaded. So you're kind of out of the snotty conditions a lot of the time. Anyway, I digress. And of course, it comes with, need not be said, that the diameter of plankton nets can come in different sizes. Typical ones are about 1 meter across in diameter. So what's going on? Our results highlight the numerical primacy of small microplastic particles in seawater, and we have found that the most abundant microplastics were between 1 and 14 microns. 60% were under 5 microns. None were larger than 53 microns. said Medina Fal. The size fraction has been totally overlooked in almost all marine microplastics surveys, which means when the plastic problem is a lot bigger than we thought. In addition, the microplastic detection method also enabled the researchers to determine microplastic particle identities and sizes from which masses were calculated. He explained this is a critical component of the work because mass-based inventories lead to more accurate and meaningful estimations of global oceanic plastic inventories, thereby providing vital information for mass flux calculations of microplastics through aquatic ecosystems. The old this ties into that concept of biological enhancement, biological accumulation, biological concentration as you go from trophic level to trophic level to trophic level. The research team reported that based on the published microplastic surveys in oceans around the globe, their work detected microplastic less than 53 microns at a rate of, 6 orders of magnitude higher than all of the combined reports. Yowzer. That translates to hundreds of thousands more microplastics. These smallest of microplastics detected in the study also contained chemicals potentially dangerous not only to marine life, but to human health if consumed significantly through the food web, going back to the bioaccumulation, bioconcentration concept. The most abundant polymers detected were polypropylene, polystyrene, polyethylene, which is consistent with the composition of plastic waste generated globally in oceans. Understanding health risks from exposure to microplastics is a vastly understudied area, and investigations are needed to understand the health impacts from microplastics of different shapes, sizes, and compositions, says Jamie Melliker, who is a professor in the program of public health at Stony Brook University, not involved in the study. Melliker's call for more public health research on exposure to microplastics includes investigation on microplastics that may enter human consumption through the seafood web, other dietary sources such as drinking liquids in plastic bottles. And of course, as Medina Fal suggests, wider research on addressing and capturing this huge problem of microplastics in the world's ocean to get a handle of how much is actually there is very challenging. Why is that? Well, the ocean is extremely undersampled, right? It's a big place. And the majority of existing data is based on towed net studies. Smaller, less buoyant microplastic particles residing below the ocean's upper few meters are almost never sampled. So that implies that most of the prior sampling for this specific problem, trying to quantify the microplastics, have been horizontals. Secondly, understanding sources of microplastic transport and final landing locations, such as the ocean floors, is crucial to explain the distribution patterns of microplastics throughout the ocean. And plastics have been found in every ocean, from the surface down to the Marianas Trench, from the equator to the Arctic. But as recent studies seem to indicate, microplastic also impacts the sequestration of carbon to depth because they serve as a scaffolding for phytoplankton and bacteria and other organisms attach themselves to, and because it's more buoyant, it doesn't sink, so it doesn't take the carbon with it. We don't get the the marine snow that we're used to seeing. So this is just, another example of more problems the plastic pollutions are posing to the ocean. And it's troubling. It gets into the food web, into the trophic levels, possibly human consumption. So, there you have it. Until next time.